Hello boys and girls, in the previous session we have understood about pollen pistil interaction, and in this session I am going to explain about artificial hybridization. See, what is the meaning of hybridization? It is nothing but fertilization, correct? So what is the ultimate product of fertilization? You get young one, correct? So artificial hybridization it's nothing but an approach for crop improvement program. So it is nothing but crop improvement program, got it? So artificial hybridization is mainly to prevent unwanted pollination in flowering plants. Unwanted pollination in flowering plants. It is normally employed by a farmer to avoid unwanted pollination of the flower, so that he can improve the crops. The crop yield will be more. Okay, so let us see how exactly artificial hybridization is done. It is done in two steps. The first step is called as emasculation, and the second step is called as bagging. Okay, so emasculation is nothing but the process of removal of the anther with the help of the forceps in a bisexual flower. See, I am very clear here. See, this is a bisexual flower. I am going to put the anther here. So these are all the anthers. Okay, so it's a bisexual flower. Okay, this is the symbol to denote a bisexual flower. Here, I don't want this pollen grain released from the anther to fall on this stigma. So I want to avoid this. You know what will happen if self-pollination occurs. I want to avoid this. How can I avoid this? Artificially. Very simple. Take a forceps. You know what forceps? You have seen it in the lab, right? You have seen that. Okay, forceps. So I am going to remove the anthers. I am going to pluck the anthers. I will remove it. So once I remove the anthers, what is left? Only gynoecium is left in the flower. That means self-pollination is impossible. So that is emasculation. It is the process of removal of the anthers from the or with the help of the forceps in a bisexual flower. So this is 100% employed in bisexual flower. Remember it. Only in bisexual flower, not in unisexual flower. You know why? Okay, so that is the thing. Once that emasculation process is done, so I am going to continue the process. And one more thing, I forgot to tell you that when exactly emasculation is done. So it should be done before a flower attains reproductive maturity. Before reproductive maturity. Remember this. Okay, so once the flower attains reproductive maturity, and if I do emasculation, there is no point, correct? Okay, so we have to do it before a flower attains reproductive maturity. At that time, the stigma is not able to receive any anthers. That's all. Okay, so the flower is young in this case, emasculation. And bagging. Bagging is very simple. I'm going to draw. Okay, so I'll continue here itself. Simple. Once the emasculation is done, we call it as emasculated flower. Only gynoecium is there. So I'm going to take a butter paper and I'll just cover this flower. You know why? Simple. I don't want, when the flower is developing, I don't want any foreign pollen grain to fall on it and pollinate it. So if a foreign pollen grain pollinate this, then whatever I have done so far is waste, correct? So I am going to cover this with a paper. What paper? Butter paper. So what is bagging? It is the process of covering the emasculated flower by a butter paper to avoid unwanted pollination. Okay, so I'm going to use butter paper, so that is bagging. So bagging can be employed both for bisexual flower as well as unisexual flower. Remember. So what is the main intention of bagging? To prevent the contamination of the stigma, correct? It prevents contamination. So what is the meaning of contamination? It is nothing but the pollination by some foreign pollen grains. That is actually the contamination. Prevents contamination of stigma. Okay, remember it. Okay, hope you can see this edge. Prevents the contamination of stigma. Okay, bisexual and unisexual we can do bagging, but emasculation should be done only for bisexual flower. So that's all about it. And once bagging is done, now you know that it attains reproductive maturity. After some time, this same flower will attain reproductive maturity. At that time, the stigma becomes receptive, correct? So it is able to receive the pollen grains. Now what I have to do? I have to bring some pollen grains which are already collected, and it has got some qualities. Okay, so these are all the desired pollen grains. I have already collected it from some other flowers of the same species, or maybe some different variety. Variety means within the species. Okay, so desired pollen grains. So now what I have to do is, I'll write here. So this is the flower, and you see the gynosium here, only the stigma is there. Now I am going to sprinkle this. Before I sprinkle, I have to remove the bagging. Correct. I have to remove this bag. So once I sprinkle this desired pollen grain, now almost pollination is done. Now again I have to use bagging. That is also called as rebagging. After sprinkling the desired pollen grains on the emasculated flower, now I have to go for rebagging. So rebagging means again I have to take another butter paper and cover this pollinated flower. So the artificial hybridization is a process which involves emasculation, bagging and rebagging to produce desired plant. Correct. So I want a desired plant. So here I am doing all this process. Hope you understood this concept. In the next concept I am going to explain about double fertilization.